So, good morning, everyone. I would like to welcome you in the second course. If I am correct, you already had a lecture with Dr. Trnka. That was about what topic? And the answer on this question is? More loudly. Change in entropy, some other answer.
Gives energy.
Gives energy, or maybe
Thermodynamics.
Or thermodynamics at the whole, or energy, you can say. So you know that this whole question, this whole simple question, has quite a difficult answer, and there are many chemical equations and many mathematical equations that can describe it. Today we will go to some other field that it's good to start with before we go deeply inside the metabolism. And this lecture is called the overview of the energy metabolism. So during this lecture, I would like to give you a basic overview. What does it mean, the energy metabolism? The majority of the lecture will be focused on drawing of the basic pathways of our metabolism. Or better to say, the position of the basic pathways in our energy metabolism. Before we will go to this, we will define some basic terms. That means, for example, what does it mean anabolic? What does it mean catabolic? What does it mean metabolism? And then, at the very beginning, we will mention some small topics, for example, about the regulation of the energy metabolism. For after this whole course, you should be able to answer several questions. Yes, you should be able to answer what metabolic pathways do we have in our body, where they are located, what's their role, that means when and why our cells are doing them, and the last is how this whole process is regulated. And this opening lecture should give you the whole overview, and then during the whole course you can say good. So right now we are discussing about this pathway, and this pathway is connected to this and to this. Yes? So if we will start with the basic definition. What does it mean metabolism? If you look in your book, you can see many definitions. Here I choose some simple definition, and that is that metabolism is a set of catabolic and anabolic processes, or anabolic and catabolic reactions. Inside the metabolism, you will meet compounds that we call intermediates or metabolites. You can say energy metabolism or intermediary metabolism. For us, this means the same, and it's the concerning all basic pathways. That means basic metabolic pathways of our saccharides, lipids, and proteins, plus the common pathways as is Krebs cycle and respiratory chain. Yes? All these things together we call energy metabolism or the intermediary metabolism. During other courses, you will speak about other metabolic pathways. They typically do not belong to the energy metabolism. For example, metabolism of reactive oxygen species, or during the inflammation processes. When we speak about catabolic and anabolic reactions, we should define what this means. As you know, when something is catabolic, that means that you, from some more complex compound, produce some simpler compound. Therefore, typically these reactions are degradation reactions, and during the catabolic processes, as the net balance, you should release some energy, and therefore they are exergonic reactions. One single reaction could be endergonic in the whole series, but the whole process, for example glycolysis, should be exergonic. Typical example of catabolic reactions and catabolic processes is, for example, proteolysis. That means the reaction where you take one big protein at the very beginning, and at the end you have many single monomers of amino acids. And as you know, we have altogether 20 proteinogenic amino acids, or 21, and all proteins are composed of them. And during proteolysis, you release these amino acids from big protein, and this is a catabolic process. On the other hand, we have anabolic reactions that are typically synthetic reactions, and the whole process should be endergonic. That means that you must give energy inside the reaction or inside the whole process to proceed. And typical example of anabolic process is opposite process to proteolysis. That means synthesis of new proteins on ribosomes. That means protein synthesis that's located on the ribosomes. You know that you take the single monomers, the amino acids, and you make there the peptide bond, forming a new long protein. Other example could be, for example, synthesis of triacylglycerols, when you take 1 molecule of glycerol, 3 molecules of fatty acids, you catalyze the formation of an ester bond, and by this you produce 1 bigger molecule of triacylglycerol. In our body, there are some processes where you simply cannot say whether they are catabolic or whether they are anabolic, due to the fact that they are so complex that one part is catabolic and from our point of view they are anabolic. These reactions we call amphibolic, that means on both sides, and typical example of these reactions is Krebs cycle. Catabolic point of view of Krebs cycle is the point of view when 2 carbons from acetyl inside the acetyl coenzyme A are degraded and you release carbon dioxide. That means from acetyl you produce 2 molecules of carbon dioxide, simply your compound is released, therefore it's catabolic. On the other hand, Krebs cycle has many intermediates inside that serve as the precursor for more complex compounds. For example, succinyl coenzyme A can give birth to the heme, that's important for the hemoglobin in our red blood cells, or alpha-ketoglutarate can be converted to neurotransmitters, for example to glutamate. And therefore we can say that Krebs cycle share both characteristics, catabolic and anabolic pathway, and therefore we call it amphibolic. Do you understand this? So if you understand this, then we will look on the basic overview of the energy metabolism. This is the basic overview. So as you see, our whole metabolism is quite simple. We have variable nutrients coming inside our metabolism. The source of them is typically diet. And here we have proteins, saccharides, and lipids. These compounds are oxidized. Therefore, we need to have the oxygen. And via the oxidation of nutrients, you produce energy and you produce waste products. As you know from previous lecture, our body conserves the energy for immediate use in form of ATP. And we call this a macroenergy compound. And it's a general form of energy for many reactions, for many transport proteins in our body. Here we have 3 basic waste products of our energy metabolism. All nutrients produce carbon dioxide and water. Carbon dioxide is released especially in Krebs cycle, and water is produced in the respiratory chain. And then here we have the urea, and urea is the waste product of the amino group or the nitrogen inside amino acids. That means that urea is not produced from lipids nor from saccharides, only from proteins, that means only from amino acids. Of course, that the intake of nutrients is not constant, and there can be some gap in the nutrient, and therefore we have some storage compounds in our body. That means that the nutrients can be converted to the storage compounds, and then these compounds in the period of starvation can be released from the stores, and they can be oxidized, giving you the energy plus the waste products. Could you name me some storage compounds that we have in our body? Glycogen, that's for what?
Glucose.
That's for glucose, that means for saccharides. And the second? Triacylglycerols, that are the main energy storage compound. And we have the majority of energy in glycogen, in triacylglycerols. In glycogen, we have the glucose molecules. We do the oxidation and the production of energy due to the fact that we have some energy demands. On this picture, you see the basic energy demands, the list of them. Why do we need to produce the ATP in our body? So we need to have ATP to synthesize some big molecules in anaerogenic processes. We need it for muscle contraction. We need it for active ion transport. And then we need it for the thermogenesis. Could you tell me what's the main energy demand for our body? So what of these four processes consumes the majority of energy in our body? Thermogenesis, not. That's, I think, at the least. Muscle contraction, typically also not. It depends how active you are. But as we look here, right now as we are sitting, muscle contraction is not the main. Thermogenesis, not. Macromolecules also not. Active ion transport. Yes, it could be for you quite surprising that this quite simple process, but it consumes more than 50% of all energy in our body. And for example, in our neurons or in the nerve system, it consumes over 80% of all energy. And it's due to the fact that, for example, the neurons, if we now focus only on neurons, their role is to produce action potential. And action potential, that means the movement of ions across the membranes. Sodium cations going inside the cell during depolarization, and potassium cations coming outside the cell during repolarization. And as I will tell you at the end of the lecture, our brain consumes 20% of all energy of our body. Yes? So active ion transport you need for many, many important processes in our body. For example, calcium cations serve as the second messenger. That means many hormones are utilizing this ion for the transmitting their signal inside the cell, and whole nervous system and whole excitatory system is based on the transport of ions across the membrane. So that's the main. Of course, the muscle contraction, here it highly depends what do you do. Yes? If you have a sedentary lifestyle, then the muscle contraction is not the main. If you are worker, miner, or if you are professional sportsman, then of course physical activity is much more important. But if you look at the typical society right now, there is not so many workers, yes, and not so many miners. Yes, majority of us have very small physical activity, unless we do it actively by ourselves. Now in 3 slides, I will give you the basic overview of the names of the pathways that we will meet today, and then that you will meet these whole curls. So on the first, we have the list of catabolic reactions proceeding in our body. First, we start with the saccharides. Basic catabolic reaction of saccharides is called glycolysis. Glycolysis is a reaction where you convert one molecule of glucose to 2 molecules of pyruvate or lactate. That means typical catabolic process. Second is called degradation of glycogen or glycogenolysis. That's the reaction where you take the big polymer glucose, the glycogen, and you produce the single monomers of glucose. Then we have lipids. Lipolysis is similar to glycolysis, only you start with triacylglycerols, and by enzyme that's called lipase, you destroy the ester bond in triacylglycerol molecule, and you produce fatty acids and glycerol. Beta oxidation is a subsequent step. That's the step where you convert molecules of fatty acids that typically are long. They have 16, 18 carbon atoms, and you convert them to 2 carbon acid active residues that we call acetyl coenzyme A. Coenzyme A itself is big molecule, but that's only cofactor. The active part has only 2 carbon atoms, that's the acetyl, and therefore the whole process is catabolic. Ketone bodies degradation, today only take into account that this exists, yes. More in detail, we will speak about it in around 2 weeks. Ketone bodies are produced from acetyl coenzyme A from fatty acids, and then their degradation is also the catabolic reaction. And last, that are proteins and amino acids. The proteins are degraded in a reaction called proteolysis, and then each amino acid is degraded in a unique pathway, and in general we can say that it's degradation of amino acids. Any question to this?
Could you repeat the short definition of lipolysis?
Lipolysis is the reaction where you start with triacylglycerols and you produce 3 molecules of fatty acids, or 3 fatty acids, and 1 molecule of glycerol. So you start with triacylglycerols and you produce 3 free fatty acids and you produce glycerol. Any other question? So now, anabolic reactions. As you will see, they are to a very big extent opposite of the previous. First is synthesis of glycogen. In this reaction, you simply take glucose monomers and you produce glycogen. That means big polymer of glucose molecules. Second is called gluconeogenesis, and gluconeogenesis is the reaction where you take some small compounds, typically 3 and 4 carbon atom compounds, and you convert them to the molecule of glucose. For example, you can start with pyruvate, with lactate, or with some carbon skeletons of amino acids, and you convert them to the glucose molecule. Then we have lipids. We call this synthesis of fatty acids. That means the reaction when you start with acetyl coenzyme A molecule, and in a series of reactions you produce fatty acids, typically with 16 carbon atoms, and then you can elongate it. Subsequent step is lipogenesis, synthesis of triacylglycerols, and that's the reverse process to this. That means you take fatty acids, you take glycerol, and you produce triacylglycerols. This reaction typically proceeds in your adipose tissue. Ketogenesis, that's the reaction where you produce ketone bodies. So ketone bodies are alternative energy substrate during starvation, and you produce them from fatty acids, yes, in a simple series of reactions. And the last are reactions concerning proteins and amino acids. That means synthesis of proteins on ribosomes, protein synthesis, synthesis of amino acids. We can produce in our body 12 of those 20 proteinogenic amino acids. That means not all of them, but only some of them. And the last reaction here is synthesis of urea. That's the synthesis of the waste product of the protein metabolism, but the whole pathway is anabolic. For you start with ammonia, carbon dioxide, that means 2 simple compounds, and you combine them together, and you produce urea, that's bigger compound. So the whole process is anabolic. Do you understand this? And now here we have the common reactions that forms the very heart of the whole energy metabolism. That's Krebs cycle. After Krebs cycle, you typically run the respiratory chain. That means the place where you consume oxygen and produce ATP. And one step ahead, Krebs cycle is called pyruvate dehydrogenase reaction, where you convert pyruvate, that's 3 carbon acid compound, to acetyl coenzyme A, and this acetyl has 2 carbon atoms. But typically, pyruvate dehydrogenase reaction is taken together with the Krebs cycle, and therefore we say that it's one big heart of the energy metabolism. But sensu stricto, this is the catabolic process. For here you release carbon dioxide as the side effect of the whole reaction. And the last, before we start drawing, are here these 3 intermediates. 2 of them you have on the board. That's the pyruvate, that's the acetyl coenzyme A, and what's the last? And what's its role in the metabolism? Its role is to transfer electrons and hydrogen protons in our metabolism. So typically, we call these cofactors of oxidoreductases, NAD and FAD. They have slightly different position, they react with different enzymes, but we can say that they help the enzymes to do their role in the metabolism. Yes, therefore they are cofactors. And these 2, NAD plus FAD, they work as the cofactors of oxidoreductases. And as you saw, the energy metabolism in the catabolic direction, that's the oxidation. And these will help us to take the electrons, to take the protons from the substrates, and they give these electrons into the respiratory chain. That is also called electron transport chain. And from these electrons, we gain the energy to produce ATP. So here we have the whole picture. So you can imprint it into your brain. And right now, we should draw this on the board. So we start with proteins. Then we have saccharides. And then we have lipids. And as you remember, we have 3 waste products of our metabolism. The first is urea. Second is carbon dioxide. And the third is water. And as you know, the thing we want to produce in our energy metabolism is ATP. So, where do we produce the majority of ATP in our body? How do we call the pathway? Oxidative phosphorylation, very good. And one step ahead, oxidative phosphorylation is called? That's the whole chain. Electron transport chain or respiratory chain. And here we can draw a mitochondria for the whole electron transport chain plus oxidative phosphorylation proceeds in mitochondria. So respiratory chain plus oxidative phosphorylation produces ATP. As the substrate for oxidative phosphorylation, you have ADP plus the inorganic phosphate, as they are coming from the solution and they are dissolved inside. What do we need as other substrates for respiratory chain? Very good. So this is the reason why we breathe in the air. For we need to have the oxygen, and the majority of oxygen is consumed in the respiratory chain. You have some other reactions also, but that's only the minor. Yes, majority of oxygen is consumed inside the mitochondria in electron transport chain, and oxygen molecule serves as the final acceptor of electrons. When it accepts electrons, it's reduced, gains minus charge, and it will react with hydrogen protons, giving you the water. Could you estimate how many milliliters of water do we produce in our body per day?
3 liters.
3 liters is too much. Around 300 milliliters, 400 milliliters. Why do you think that this is important? Or do you think it's important? That was a bad series of questions. You know, typically this is quite small amount. For you should per day drink 2 liters of water, yes, in all sources. But there are some people that have problem with water metabolism and water balance in our body. There are typically people that have the kidney failure, some kidney diseases. For kidneys serve as the main regulator of the water and ion metabolism of our body. And as you know, some of these people must attend the dialysis, and they attend it, for example, 3 times a week. And they must take care of the amount of water they drink per day. Plus, the doctor must take into account that every day the normal metabolism produces around 300 milliliters. If you would not take into account this water, then it can bring the disbalance to the patient. Yes, so you must know that some metabolic water is produced every day, and if you count it per 1 week, you produce around 2 liters of water. Yes, so it's not a small amount of water. The last substrate for the respiratory chain are these reduced cofactors. That means NADH plus FADH2. As you will heard, different enzymes have different cofactor, but in the whole metabolism you produce both. And also these cofactors react with different complexes inside the respiratory chain. They work as the source of electrons that feeds the electron transport chain with electrons, that means with energy. What's the main place where do you produce reduced cofactors in your body, in our metabolism? It was correct. Mm-hmm. Krebs cycle, that's the main place where you produce reduced cofactors. In 1 spin of Krebs cycle, you produce 3 molecules of NADH plus 1 molecule of FADH2. Yes, per 1 rotation, that means per 1 molecule of acetyl coenzyme A that's coming inside the Krebs cycle. The second part is oxalacetate. That's 4 carbon atom compound. Acetyl coenzyme A, the working part, has 2 carbon atoms, and therefore the first intermediate has 4 plus 2. That means 6 carbon atoms, and we call it citrate or citric acid. And therefore the alternative name of Krebs cycle is citric acid cycle. And this is 6 carbon atoms. Since we need to return during the whole cycle to the 4 carbons of oxalacetate, we must, during the whole process, release 2 carbon atoms. And both these carbon atoms are released in form of carbon dioxide And therefore, per one rotation, we release 2 molecules of carbon dioxide. And this is the reason why we breathe out. That means, where do we produce carbon dioxide? Yes, it's a very big producer of carbon dioxide, this Krebs cycle. It's not the only, but it's a very important producer of the whole thing. As the result of Krebs cycle, you have the reduced cofactors that feeds the respiratory chain, and you produce the waste products, carbon dioxide, and also you produce there some small amount of energy directly in the whole Krebs cycle in form of GTP. How do we produce acetyl coenzyme A in our body?
Production of pyruvate.
Oxidation of pyruvate.
It's oxidation of pyruvate. So we take pyruvate. As you remember, pyruvate has 3 carbon atoms. And the whole reaction is oxidative decarboxylation. And that means that you produce carbon dioxide. So this is the second important place where you produce carbon dioxide in your body. So pyruvate dehydrogenase reaction, abbreviated as PDH, when pyruvate is converted to acetyl coenzyme A. This reaction is, under the conditions of our body, almost only irreversible. That means that you can convert pyruvate to acetyl coenzyme A, but the opposite reaction is very difficult to proceed in our body. So we can say that is almost fully irreversible in our body. And that's one of the reasons why you can convert pyruvate to acetyl coenzyme A, but it's very difficult to convert acetyl coenzyme A to pyruvate. And therefore also acetyl coenzyme A to saccharides. And it's very, very limited ability to do this. Yes, it's almost fully irreversible. But as you know, nothing is absolutely irreversible. Yes. How do we produce pyruvate?
From glucose.
Yes, from glucose. The reaction is called glycolysis. And as you know, glucose has 6 carbon atoms. And that means from one glucose, you produce 2 molecules of pyruvate. The reaction is called glycolysis. It has many, many steps. And during the energy metabolism, it will be one of the first pathways that we will do. The glycolysis can end with 2 basic products. The first is pyruvate, that is subsequently oxidated to acetyl coenzyme A, and it enters the Krebs cycle. That we call aerobic glycolysis, and we do this if we have oxygen. If you do not have oxygen, then you do not produce pyruvate as the final product, but you produce lactate. Lactate has also 3 carbon atoms. But here you have keto group, and here you have hydroxy group. That means that this is reduction, and in that direction is oxidation. This whole reaction is fully reversible, and you can convert lactate to pyruvate and pyruvate to lactate. It only depends on the availability of oxygen and on the whole redox state of the cell. Opposite reaction from pyruvate to glucose is called gluconeogenesis. And the whole gluconeogenesis consumes some amount of energy due to the fact that you convert simple compound to more complex compound. What's the second source of acetyl coenzyme A in our body? Here we have saccharides, and we have some other sources.
Lipids.
And what part of lipid molecule?
Fatty acid.
Triacylglycerols and the fatty acids. Yes. So as you know, in the stores, we have triacylglycerols, and they can be degraded by lipolysis to glycerol and to fatty acid. And we say free fatty acid for it's not esterified. Glycerol has 3 carbon atoms. Fatty acids typically have 16, 18 carbon atoms. In the process of beta oxidation, you convert fatty acids into coenzyme A molecule, and it's the stereotypic spiral pathway where you repeat several steps and you still repeat them, and in each spin you produce molecule of acetyl coenzyme A, plus you produce quite big amounts of reduced cofactors of NADH and FADH2. Both these are produced in the beta oxidation. Opposite reaction is called synthesis of fatty acids. You start from acetyl coenzyme A and you produce the molecule of fatty acid. It's also the spiral process, but here I make the single direction, yes, to be more clear. Yes, but it's also a repetitive stereotypic process, only catalyzed by different enzymes. Yes, but as you will see, when here it's oxidation, here will be a reduction. Yes, so it's to big extent opposite process. Glycerol, since it has 3 carbon atoms, typically can be converted to glucose, or better to say, it can join the metabolism of saccharides. For here, the whole glycolysis, or not whole, big part of glycolysis is at the level of 3 carbon atom compounds, and therefore glycerol can be part of glycolysis or gluconeogenesis. Yes, that depends on the state of the cell, but typically it serves as the substrate for the synthesis of new glucose molecules, since it joins the gluconeogenesis pathway at the level of dehydroxyacetone phosphate. Yes, the basic triose with 3 carbon atoms. Any question to this, to the lipid part of the metabolism?
What's the interaction of the degradation of lipids?
This is called lipolysis. This is beta oxidation. And this is the utilization of glycerol. Yes? And if you want, here we have the opposite reaction. So this is the synthesis of fatty acids. This is also reversible, so you can synthesize the glycerol. And then here the opposite reaction is called lipogenesis. That means production of triacylglycerols. We said that triacylglycerols are the storage compound. Glucose has also the storage compound. The storage compound is called glycogen. And glycogen molecule is produced by the synthesis of glycogen from glucose monomers. And in glycogenolysis, you produce back glucose. Yes? So synthesis of glycogen and degradation of glycogen. Why do we have here only glucose and no other saccharides in the whole scheme? Why do you think?
Extracellular, they broke extracellular.
They typically intracellular, they are converted. The fact is that glucose has the central position due to the fact that all monosaccharides can be converted to glucose and monosaccharides can be produced from glucose. Yes? We will speak about specific pathways. With Dr. Kvasnitsova, you will speak about the utilization of galactose, utilization of fructose, and about the pentose phosphate pathway, where you, for example, produce ribose that's necessary for the DNA structure. But all these monosaccharides can be converted to glucose or can be produced to glucose. Yes, that means that the glucose molecule itself has the central position, and here you can say fructose, galactose, ribose, but when you want to degrade them, you must return to the glucose. Last part of the scheme, that are proteins. So the first reaction when you want to oxidize proteins, that's the proteolysis, when you convert them to amino acids. This reaction gives you the monomers, and as you know, typically in proteins we have proteinogenic amino acids. Some of them can be modified, yes. We have some, for example, hydroxy derivatives of proteinogenic amino acids, but you have only those 20 that should be typically in the proteins. Each of these amino acids has the unique pathway how to be metabolized. Some pathways are very simple. Some, for example, from tryptophan are very long and very difficult. Today, we can make one simplification, and the simplification is that amino acid means two things. First, that's the nitrogen inside, so we can say that it's amino group that's inside. But it means all nitrogen that's inside the amino acid. And second, that's the rest of the molecule that we call carbon skeleton. That means that's the body consisting of carbon, hydrogen, oxygen inside the amino acid, and the other part is only the amino group, or in general the nitrogen inside the amino acid. And these two parts of amino acids, they have different fate in our body. The amino group is typically converted to the urea. How do we call the reaction? Where do we convert it? Urea cycle, or the alternative name is ornithine cycle. For ornithine is one intermediate of the whole cycle. So in ornithine cycle or urea cycle, you convert the nitrogen from amino group into the urea, and here we use some amount of carbon dioxide. Yes, for urea has carbon, oxygen, nitrogen, hydrogen, and the carbon is coming from carbon dioxide. The result of this whole process is urea, the last waste product of our metabolism, and urea cycle typically proceeds inside our liver cells. And liver cells must degrade the amino acids that we have in the diet or our own amino acids. What can we do with the carbon skeleton? How do we join it with this metabolism?
Change the acid to covalent.
That's one possibility. Some amino acids can give us acetyl coenzyme A. What else?
Pyruvate.
So pyruvate, here was pyruvate. Do you know, for example, what amino acid is transaminated to pyruvate in single-step reaction?
Alanine.
Yes. Alanine is transaminated to pyruvate. For alanine has 3 carbon atoms. So it's single transaminated to pyruvate. And the enzyme is called ALT, alanine aminotransferase. And all of you know it, since if you look into your lab results when you go to the GP, ALT is one of the liver enzymes that assess the state of the liver, whether there is no liver damage. So here, single step to transamination, and other possibility is, for example, that they join the Krebs cycle. For example, aspartate is transaminated to oxalacetate, glutamate is transaminated to alpha-ketoglutarate. Yes, all 20 amino acids can join the metabolism somewhere, and we will to some extent speak about it during the respective lecture. But today, it's enough for to know amino group, carbon skeleton, amino group, urea and urea cycle, carbon skeleton joins the rest of the metabolism. And the last, what I should put here is the interconnection between urea cycle and the Krebs cycle. Since these two pathways are joined together, yes, some part of urea cycle proceeds in mitochondria, the same, the whole Krebs cycle proceeds in mitochondria, and they exchange their intermediates inside to be fully effective. So, any questions to proteins or to the whole scheme?
Yes, can you just repeat this here?
This?
Yes.
There's the fact that the urea cycle typically works in concert with the Krebs cycle, for they exchange their intermediates at two sides. Yes, fumarate, aspartate that are exchanged one pathway to the other, and therefore working together to produce the urea as the waste product. Yes. Any other question, or do you want to have a break? Break. That's excellent. I think so also. Now we have 3 smaller topics, and these topics should give you only some small overview what also you will learn in this course. Yes, right now the main thing what you should learn this week is this. Yes, to understand what's the basic connection in the pathways, and then this is only small tasting on the things that you will learn also. Yes, you do not have to memorize it, for you will have it during the whole year, during the whole course. You will listen or you will hear that something is happening in liver, something is upregulated or downregulated by insulin. So today I want only to tell to you that in different organs, by different hormones, we regulate different pathways. Yes, but you do not have to memorize it right now, only try to gain the bigger overview. We have many organs in our body, as you know, and these organs, they typically have some specific position in the energy metabolism. Some are typical complex organs, as for example liver, where proceeds almost everything. Then we have, for example, brain or adipose tissue that has one big significant role, and the other roles are more minor. If we start with the liver, inside the liver you do almost everything. Yes, almost all enzymes, all metabolic pathways can to some extent proceed in liver. There are some exceptions, but this scheme you can say that as whole exists in the liver. That's also one reason why we do not have artificial liver. For to have some instrument that copies all these functions of the liver, it's almost impossible. For there is so many reactions with so many regulations that we are not able to produce artificial liver since far. You know that we have artificial heart. It's called HeartMate. It's the thing that produces the continual flow of the blood. Of course, it's not ideal, but that's the main role of heart, to pump the blood through your body. So this you can do for heart, but here it's very difficult to say what's the main role of liver inside the whole metabolism. Here you have something what happens in the liver. So as the whole, we can say that it maintains the homeostasis. That means the steady state of our body. Yes, it's able to upregulate or downregulate pathways to increase or decrease production of some compounds, or it can do the transformation of one compound to the other. For example, saccharides to lipids, proteins to lipids. These conversions typically proceeds inside the liver to some extent. Also inside the liver you store the glycogen. Inside the liver you have 80 to 100 grams of glycogen, and that means that you have there also the glucose inside. And of course inside the liver you produce also waste products. It's a very important organ for detoxification of your body from your own waste products, or it's doing the detoxification of xenobiotics. That's, for example, drugs, that's pesticides, that are things put inside the food as the conservants. Many of them are metabolized inside the liver and then excreted in the bile. That means all these functions proceed in one organ, and it's very difficult to replace all of them. As the small overview, we can say what happens there during the starvation and in well-fed state with the saccharide metabolism. So as you know, during the day, you have periods when you have a meal, and then you have the free GI tract. One of the metabolites that you produce is glucose, and the glucose should be in your blood in some constant concentration, in some constant range. And liver cells work as the organ, as the executive organ of our body that is able to keep the glucose plasma level stable, and we call this the glucostatic function. And if you look here at the scheme, we also have the answer how the liver cells are doing it. So if you eat some saccharide-rich food, for example pasta, for you know that pasta, that's a lot of sugar inside, and you have the rise of plasma glucose, the typical reaction that proceeds inside the liver is the synthesis of glycogen. That means you take the glucose molecules from the blood and you incorporate it into the glycogen inside the liver. Then, if you have the excess of glucose, then you can oxidize it in the glycolysis, and you can fully oxidize it in the Krebs cycle, respiratory chain, or you can convert saccharides to acetyl coenzyme A and then produce fatty acids and triacylglycerols. All these things proceed inside the liver, and liver cells are the first cells that meet the nutrients from intestines after the intestinal wall. For all blood from intestines come inside the liver through vena porta. On the other hand, if you have lack of glucose, that means that you are fasting, you have no meal in your GI tract. In that time, the liver cells must do everything to keep the blood glucose level stable. And what they can do is to release the glucose from glycogen molecule by the glycogenolysis, or to produce glucose from pyruvate or amino acids in the gluconeogenesis. And since you have only limited amount of glycogen, then after 1 day of starvation, you are totally dependent only on the gluconeogenesis. And liver cells are the main place where you run gluconeogenesis. Do you understand this about the saccharide? And you will hear much more during the other lectures. Here, you have something about the amino acids. I told to you that ALT, alanine aminotransferase, we call liver enzyme, and that is assessed for the screening whether you have damage of liver. And we can say that the whole process of transamination, deamination, that means this process of degradation of amino acids is to big extent located inside the liver. That means that you have here amino acids, you produce from them carbon skeleton and urea as the waste product. The whole urea cycle is inside the liver. That means that if you are missing a liver, that means if you have liver problems, some liver disease, one of the results will be the disbalance of the whole amino acid metabolism. And maybe some of you know the term liver encephalopathy. That's the state if you have liver cirrhosis, for example, due to hepatitis or alcoholism. And the result is that the accumulation of these amino groups, and they will be in the form of ammonia or ammonium, that means NH3 or NH4 plus. They lead to disturbance of your brain, and typically these people can die due to it. Yes, for the ammonia is toxic to your body, especially to the brain, that has a very big energy demands. And when this is not detoxified in liver, it will affect your brain, and it's a very serious condition, the liver encephalopathy. Other thing that we have mentioned here is that liver cells are the cells that produce your plasma proteins. And you know that one important component of plasma proteins are those proteins that stop the bleeding. Yes? We call this coagulation cascade. And one of the results, if you have damage of liver, is that the patients can bleed out, since they have the problem with the blood coagulations, for they are not able to produce the plasma proteins. And now you can imagine what we must replace in the patients that have problems with the liver, and it's almost impossible to do it fully. Yes, to replace all these important functions. Any question to liver? Kidneys. Kidneys have one specific position. How would you define the role of kidney in your body? What do they do?
Pressure in the blood plasma.
Yes, so they produce urine, they keep the isotonic condition in blood plasma. Filtration, that's at the very beginning of the urine production. And then there is intensive transport of ions. So we can say that inside the kidneys, you do very, very intensive transport of ions and transport of water. The primitive urine, that means the urine after the filtration, per day you produce 180 milliliters. And the definitive amount of urine is around 1.5 liter. That you can say that 99% you must transport back to your body. And of course, this needs to have a lot of energy. And therefore, it's not surprising that energy demand of kidneys is very, very high. And they are very important consumer of the energy from the whole body. Yes? The density of energy that must be produced and utilized inside kidneys is very, very high. To feed this, they can use almost everything. Yes? We will speak about it more in detail next year, but right now you can say that the oxidation is there very intensive and they can utilize almost everything. What's the specific role inside the kidneys is that kidneys offer you alternative way how to get rid of this amino group. Urea is produced in liver and is excreted by the kidneys, but that's only the passive process, yes, that it's in the urine. But our kidneys can produce the ammonium and put it inside the urine also. If you are healthy and in standard basic condition, around 95% of amino nitrogen is transported or converted to urea, and only 5% to ammonium. But in some specific conditions, the kidneys can change this ratio to 50 to 50. That means that they excrete a lot of ammonia by themselves and they do not wait till the production of urea. And that's here that many amino acids can be also to some extent metabolized inside the kidneys. And as one other effect is that kidneys, that's the second place where you can produce glucose in gluconeogenesis. So that means this reaction not only in liver, but also in kidneys. So any question to this? Now muscle, here the story is very, very simple. You know what muscle cells do. So could you say it in one sentence?
Contract.
That's true, contract. And how do they do it? Or what's the energy process behind? Better to say. They convert two types of energy.
Myosin and actin.
That's true. Actin and myosin we call molecular motor. So the question is, what's the role of molecular motors in our body? That's true, but not only muscle contraction, also, for example, movement of vesicles in our cells, movement of mitochondria in our cells, division of cells. So the basic process, if we return to the previous lecture, is that molecular motors convert the chemical energy, typically ATP, to the mechanical energy. In the macroscopic point of view, we have big muscles, and these muscles use ATP and produce the movement. That's their position. And therefore we can speak, therefore we can move our limbs and all other functions. That means that they must have very big amount of ATP, and here you have the list of compounds what they can use for the ATP. So as you see here, they can use many molecules to gain the ATP. And one of them you know from the previous course, from the practical trainings, where you assess concentration of creatinine in urea, and the creatinine comes from creatine, and inside the muscle it's in the form of phosphocreatine. There's a macroenergy compound that regenerates the ATP. And it's a very short-term but very effective way how to give new energy to ATP. For as you know, all of us can decide in one moment to run, and till the heart reacts to this and accelerate the heartbeat and the frequency, that's very long. Yes, and therefore the muscle cells must have some instant source of energy, and creatine phosphate is one of them. Other thing that you should remember, and here it's only one thing what you should know from this slide, is the branch chain amino acids. What amino acids do they are? Could you tell me their names? Branch chain. And these amino acids are the first phases of their degradation located inside the skeletal muscle. Not in liver, but only in skeletal muscles. And we will return to them when we will speak about their degradation, and we will speak about it for they have very unique position in the situation when you have problem in liver. For if you have liver cirrhosis, you have problem with degradation of amino acids. But these amino acids can be the first part inside the skeletal muscle, and therefore they are a very good way how to help and how to give nutrition to the patients with the liver problem. For their metabolism is not so heavily affected in the patients or with the liver failure. And all these other things we will mention on the lecture. And now two last tissues. First, adipose tissue. So what's the role of adipose tissue? What do you think?
Keep the heat.
So first, isolation, that's true. Then second, that means conserve the heat in our body and protect our organs. For example, heart, kidneys, they have the adipose tissue around. What else? That means store triacylglycerols, that means store energy. Yes? In adipose tissue, we have the majority of energy storage in our body. The glycogen is very small amount if you compare it to triacylglycerols in adipose tissue. And as the effective store, they must be able to do two things: to take the compounds to the stores and then to release them. And as you know, we call this lipogenesis, and we call the opposite process lipolysis. That's one role of adipose tissue. Second role is that adipose tissue, as we know right now, is the biggest endocrine organ of our body. It produces a very big amount of signaling molecules that we call adipokines. That means hormone produced inside adipose tissue. And we know that these adipokines has not insignificant role in the civilization disorders, as for example type 2 diabetes, high blood pressure, and the whole metabolic syndrome. So to have adipose tissue is necessary, but you must have the proper amount of adipose tissue. If you have proper amount of adipose tissue, they do their function and produce the beneficial signaling molecules. If you are obese, the adipose tissue will start to start the signaling that will lead after many, many years to the development of the civilization disorders. That means without adipose tissue, it's not to be. And if you do artificial model of animal without adipose tissue, the animal is absolutely not healthy, yes? But to have a lot of adipose tissue and to be obese is also not healthy. So it has very significant role in the metabolism, the storage of energy, and to do the signaling inside the energy metabolism. And the last organ is brain. So there's the organ where you are able to listen to me, where you are able to communicate, where you are able to write poems and all these nice things. And here you have the balance. 2% of body weight and 20% of energy expenditure. That means the whole energy demand. And what's missing here is 25% of oxygen. So it's very, very big consumer inside our metabolism and very big consumer inside as the point of view of oxygen. And one other bad thing to the brain, if we return to skeletal muscle or to kidneys, they are also big consumers, but they can eat almost everything. Not for brain. For the brain is highly strictly dependent only to glucose. And per day it was calculated that it consumes 120 grams of glucose. There is only one exception, and that's the exception when you are in long-term starvation. Then it adapts, and the brain takes only 50% of this amount of glucose. That's 60 grams of glucose, and the rest is covered by ketone bodies. But what you can remember right now is that brain consumes almost only glucose, and that means that the gluconeogenesis during the long-term starvation you run to feed your brain. Not only brain, also erythrocytes, but the brain is a very big consumer of the glucose, and therefore you must do so intensive gluconeogenesis. Without the glucose, the brain is starving, and we can say that for the brain, the hypoglycemia is the same as hypoxia. So if you have low blood glucose level, the brain will simply switch off also, yes? And if you have patient with long-term hypoglycemia, it can be the same as if you have the patient with long-term deficiency of oxygen. So there can be the brain death only due to the fact that you do not have glucose in your blood. Any question to this? Good. So no question. Then the second short topic are inter-organ metabolic pathways. We right now name some organs, and these organs are typically connected by the whole series of pathways. That means that through your blood, you exchange the metabolites, and as you will see during this course, the organs communicate each other not only by signaling molecules, but also by sending the metabolites through the blood. One of the examples of these metabolic pathways is called Cori cycle. Cori cycle is a very simple pathway that proceeds only in this part of the metabolism. The Cori pathway is the pathway that couples together your liver cells and your red blood cells. When we spoke about this, I told to you that lactate is produced during anaerobic glycolysis. That means when you do not have oxygen. It's quite frustrating for erythrocyte that erythrocyte can do also only anaerobic glycolysis. Erythrocyte has a very big amount of oxygen inside. That's true, for there is a lot of hemoglobin. On hemoglobin you have a lot of oxygen. But unfortunately, it cannot utilize the oxygen. Do you know why? The erythrocytes have no mitochondria, and therefore, no respiratory chain, no oxidative phosphorylation, no Krebs cycle. Then unfortunately, the only source of energy is the glycolysis, where you produce 2 ATPs, and the pyruvate must be converted to lactate, and the lactate is released into your bloodstream. What's the problem with the lactate is that glucose is neutral compound, no effect on pH, but lactate is the salt of acid. That means that here you have lactic acid and lactate. If you have accumulation of lactate, you have decrease of body pH, and the border when you can live is around pH 6.9 or 7 of your blood. That means that when the erythrocyte produces a lot of lactate, it can kill you. Fortunately, we have the second organ, and that's liver. And the liver cells take this lactate produced by erythrocytes, and they are converting the lactate to pyruvate, and then in the gluconeogenesis, they convert it to the glucose. And this inter-organ metabolic pathway is called Cori cycle, for it was discovered by Gerta and Carol Cori, and who got the Nobel Prize for the whole saccharide metabolism discoveries. Do you know where these two Cories were studying? That's true. In Charles University, this was the German part, but they were graduating in year 1920. So maybe you will also gain the Nobel Prize. Yes? Their biggest discoveries, of course, were done in other countries, for there was the problem with the regime, for they were Jews, so they cannot stay here, but they were our graduates. And they are the only Nobel Prize winners in medicine from Prague. Yes? So we like them. So do you understand the Cori cycle? Yes, very simple pathway. We will speak about it later, but only here to show to you that you have two organs. One part is done in one organ, other pathway is done in the other organ, and in combination, they let the whole story proceed. This is the simpler scale. And the last is the regulation. So I think that all of you know that the chemical reactions in our body proceeds with the help of catalyzers that we call how? How do we call the biocatalysts? Yes, enzymes. And you know that we have two basic groups of enzymes, the normal one, and then the one that we call regulatory enzymes. Regulatory enzymes are those enzymes that control the pace of the whole reaction. And why they are regulatory enzymes? That due to the fact that they are regulated by some signals coming to the cell. And therefore, you must have some signaling molecule that affects the cell metabolism. And inside the energy metabolism, we have two groups of hormones, that means signaling molecules, that control our energy metabolism. The one group has only one member, and that's insulin. Second group has more members, and we call them counter-regulatory hormones. Do you know the name of counter-regulatory hormones? That means the hormones that act against the insulin. Yes, one is glucagon, and we have 2 or 3 other. Then we have a group of glucocorticoids, that means hormones produced from the suprarenal gland, by the cortex of the suprarenal gland, and the member of them is, for example, cortisol. If you do the first aid, then very often you have the hydrocortisone, that's the derivative that can help you if you have the asthmatic seizure or if you have some other allergic reaction. And last, what we can write here is adrenaline, or as you have sometimes in your books, epinephrine. or in form of noradrenaline and norepinephrine. Adrenaline is the hormone produced in the suprarenal gland, in the medulla of the suprarenal gland, and noradrenaline is the neurotransmitter produced by the sympathetic nervous system. And the last that you can say to some extent is the growth hormone. But these three, they are the main control regulatory hormones, and if you should name only one, then name the glucagon. All these hormones regulate our body energy metabolism, and you will, during this course, heard that glucagon is producing some second messenger in the cell and affecting this regulatory enzyme. Or insulin is deactivating this regulatory enzyme. If we make a simplification, and what you can take today is this basic picture. Insulin is the hormone that's typically produced after a meal. That means in well-fed state. That means when you have nutrients in your GI tract and you want to utilize them. And therefore, insulin is very strong anabolic hormone that produces triacylglycerols, that produces new proteins, and that produces glycogen. And also, insulin is destroying glucose molecules. That means that typically insulin lowers glucose that you have high after the meal to feed all other anabolic processes. After a meal, you have 40 times higher concentration of insulin than the concentration of glucagon. Therefore, you have this picture. Yes, you have a lot of insulin, and it's activating the anabolic processes and lowering the blood glucose level. On the other hand, if you have no nutrients in GI tract, if you are in a fasting state, then these control regulatory hormones are in favor, and their role is very simple. Their role is to give energy to the body from the storage compounds. That means that they are catabolic hormones. They cause the degradation of proteins, degradation of lipids, degradation of glycogen, and they are increasing the blood glucose level. Or better to say, they are keeping it at some stable level. What can be quite surprising for you is that even after a meal, in your blood plasma, you have more insulin than glucagon. The ratio after a meal was 40:1 for insulin, and in fasting state, you have 3:1. But this big decrease in the amount of insulin is enough to totally switch the whole metabolism. You can say that glucagon is more effective in that time. Do you know how do we call the state if the ratio is 0:1? That means if you have no insulin in the blood and only control-regulatory hormones. And what type of diabetes? Type 1 diabetes. And is it good for our body? Not. Not. For in that state, you have total catabolism. Everything is catabolic, and you have very high glucose. And without proper treatment, you will simply die. Yes? And before discovery of insulin, in the 20s of the 20th century, if you were parents of children with diabetes, you know that till 1 year the child will be deaf. Yes? For the control-regulatory hormones, there are so severe catabolism that you have no way how to stop it, only to give the insulin to the patient. Yes? No other anabolic hormone is able to stop these hormones. And therefore, you must have very sensitive balance between the insulin and the control-regulatory hormones to have the proper pace of the metabolism and to change the signaling at the right places. So, do you have any question to this? Here you have the list of the effects, yes, of the insulin and glucagon, but you will heard about it during the whole course.